Hi guys. So in this video, I'm going to cover cell membrane permeability as well as some of the mechanisms of cellular transport. Okay, so this is for lessons 2.5 through 2.9, covering all the topics in there. So it might be a little bit longer of a video overall, but if you average it out over 4 lessons, it's going to be like a shorter video than usual, I think. We'll see. So, to begin this storyline, we have to go back to the structure of the membrane and look at the phospholipid bilayer, right? And so, in this beautifully rendered model that I've made here, we have the polar heads of the phospholipids on the extracellular and intracellular sides. And then we have this non-polar region of lipid tails, and we have a protein channel. So that's what these structures are, beautifully color-coded. Right? So we have the hydrophilic region, we have the hydrophilic region, and then we have this hydrophobic interior. Okay? And so when we look at the way that molecules are going to be going across this membrane, this structure is critical to that function. Right? If you look at it just proportionally, you'll see that the hydrophilic size areas are much more narrow than this overall hydrophobic area, right? And you'll also note that there's spaces in between phospholipids. And something to keep in mind is that these phospholipids are always moving, right, in 3 dimensions across this membrane, right, around this membrane. And so sometimes they're really close and sometimes they're further apart. And depending on how close or far apart they are, different sized molecules can squeeze through. Right? Because that's a lot of times what's happening. It's like having a hole. It's like having a door or windows in the wall, right? Like if I open my window up pretty wide, a bug can come in. If I open up even wider, maybe an owl can come in, right? And so depending on how big the gap is, different molecules can get through. And when the molecules are moving across the membrane, they are moving by the principles of diffusion. Right? And so diffusion is the trend of molecules to move from an area of high concentration, where there's a lot of those molecules, to an area of low concentration, where there's less of those molecules. Okay, and that process is driven by random movement of molecules. So if there's a lot up here and they're moving randomly in any given direction, because that's how molecules move, they don't have a design about which way they want to go. They just go, right? They tend to bounce off of each other a lot, right? And so they'll bounce, 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 and they'll, by average, move towards the low concentration by happenstance. Whereas if you have a few down here, they could be moving in any direction. They might go towards the high concentration. Any given molecule, if you look at one singular instance of a molecule, isn't going to necessarily obey the rules of diffusion. It's a law of averages, right? And so this one down here in the low concentration might go up. Could happen. Happens, right? But just by average, it could also go that way, it could go that way, it could go this way, it could go that way, right? And so it's less likely that it's going to end up top than is one of these coming down below, right? And so molecules move from a high concentration to a low concentration naturally without any effort on the cell's part. And so if I have a molecule, let's look at this yellow molecule. Okay, this is representing a small nonpolar molecule. Okay, and it happens to be floating around down here. Right? If it comes up into this region right here, right, it can squeeze through the polar area because the polar area is kind of narrow. So it's just like, boop, and then it's in the yellow area, and it's cool with it being in the yellow area, right? And so it's floating up, floating up, floating up, floating up, floating up, and then it gets to this purple area, and it's like, boop, it gets through, okay? Because it's narrow enough that it doesn't matter. All right. As long as this molecule is smaller than the gaps between the phospholipids, it can just go right through. Say bye. Okay. Same storyline for the purple one here. This is a polar molecule, right? And so it comes up and it goes to go through and it gets to the polar area because it's cool with that, right? And then it hits this giant, chunky, non-polar region, and it can't get through because that's just too much space where it's not liked. And so it gets rejected, right? So it goes up, and it's like, I can't get through here. And so it goes back down, okay? So this is a polar molecule or an ionic molecule, right? Anything with a charge or a partial charge can't get through the membrane, not like that. Now, in real life, in AP Bio life, the reality is that sometimes, the gaps between the phospholipids are wide enough that polar or ionic molecules can sneak through. They just get by. They're like, bye, I'm out. Boom, prison break. It happens. It doesn't happen reliably enough where a cell can count on that to be a mechanism. Right? It doesn't happen very often. So like we tend to say it doesn't happen just as a rule, like we pretend it never happens. Right? So we say that polar or ionic substances cannot cross the membrane through simple diffusion, where small nonpolar molecules cross the membrane through simple diffusion according to the concentration gradient. Okay. Now, if we look at these larger molecules here, right, this one's purple, so it's polar or ionic, and its size really doesn't matter because we already established that even the small one can't get through, right? But just for fun, I'm going to say, it tries to go through, gap's not big enough, I can't get in. Doesn't go through. Bye. Okay. And then we have this non-polar large molecule, right? So theoretically, it should be able to pass through this based on its non-polarity. However, it's too big. It's bigger than the gap between the phospholipids. Right? And that's it. Like it comes up and it just doesn't fit through the holes. And so it can't go in. Nothing mysterious about it. Pretty straightforward. Right. And so when you have larger molecules like this one that cannot pass through the gaps of the membrane, you need a protein channel to allow these things to pass. Okay? And so these larger molecules can go through protein channels. Okay? These small polar or ionic molecules can go through protein channels, okay? Depending on the design of the protein channel. And so the protein channels are going to have different shapes with different properties, and they'll allow different things through depending on the protein channel. Right? And so this might be a protein channel that has some really strong polarity inside of it. Right? So the shapes inside might be really polar. And so if this big polar substance tries to go through, it's good, it's cool. Goes through, no problem. Whereas if this non-polar one tries to go through, it's going to get rejected by this protein channel and it won't be able to pass. Right? So protein channels can be very specific, they can be kind of specific, or they can be very general, depending on their properties. When a molecule is moving through a protein channel, but still going from high to low concentration, we call that facilitated diffusion. To facilitate means to help. In this case, the protein is helping the molecule cross. Okay? Still doesn't cost the cell any energy. And this is good for polar substances, ionic substances, and large molecules. All right. Sometimes, because the cell needs to do livity things, it wants to move molecules against the concentration gradient. Right? Like maybe there's a really, really cool molecule and it already has a bunch of it, but it wants even more. And so it wants to suck in as much of this molecule as possible. Okay? That cell is going to use active transport. And active transport requires an investment of energy on the cell's part to actively move this molecule against the concentration gradient. So it's bringing the molecule from an area of low concentration to an area of high concentration, going uphill. Okay. And so this mechanism makes use of a protein channel, right? And this protein channel is going to be shaped specifically for whatever that particular molecule is that the cell is trying to move. And so in this model, I have a rectangle-shaped molecule, and it fits into this rectangle hole here, right? Clicks on in. Okay. So this rectangle is an area of low concentration. We want to move it to the area of high concentration. So this protein channel is going to associate with the molecule, clicks on in. Okay. That's not enough to move it yet. I need to change the shape of this protein channel to actively push the molecule, right? In order to move something, you need to change shapes, right? So this protein channel needs to change shape. Now, the way that the cell is going to facilitate this change in shape is through the use of ATP, adenosine triphosphate. So I got my A with my 3 P's on it. Right? And this is what we think of as our energy molecule, right? ATP is our cellular work energy molecule. And this is how ATP works as an energy molecule. I'm going to show you right now. It's pretty simple, actually. So my A has 3 P's stuck to it. Okay. One of those P's is going to come and associate with a domain of this protein. Okay. That P is going to stick on that domain. That's going to change the shape of that domain, okay, which is going to result in a chain reaction of shape changes. Right? So I change the shape here in the protein. It's going to change the shape here, going to change the shape here, going to change the shape here, going to change the shape here. Okay? And that P gets ripped off. The ADP now, adenosine diphosphate, floats away. Bye. Right? And so we've changed the shape of the protein channel to the point where it pushed this molecule up to the other side of the protein channel. Right? So it just changed shape a bunch of times. Molecule goes up. Okay? My P is still stuck on here. Then my P falls off. My protein channel goes back to its original shape and lets go of this rectangle, and it floats away. Bye. So this rectangle has now moved from an area of low concentration to an area of high concentration intentionally by the cell by using the energy in ATP, right? Through conformational, not conformational, like I'm confirming something, but conformational, like I'm changing the shape of something, right? And so now I got my little ADP floating away. It's going to go off, get back to the mitochondria, end up getting its P shoved back on. It becomes ATP again. Whole process goes over and over again. Cool. All right. Now, in my very simple model, I was only moving the one rectangle molecule very often in order to increase efficiency. These pumps, which is what these protein channels are called, can move more than one molecule at a time. So there's paired molecules that either are going in the same direction together or they're going in opposite directions. And as the shape changes, right, it will grab both of them and move them together. Saves energy. Yeah. A really good example of that is the sodium-potassium pumps that play a huge role in the way that our muscles function and our neurons function. Right. And so we will talk about those probably when we talk about the nervous system. We'll look at how that actually works. Okay. And then we have the story of osmosis. And osmosis is, simply put, the diffusion of water. Right? And it only has its own name because water is so ubiquitous across all of life here on Earth. Right? And water in and of itself is polar, right? So if you just looked at water and you didn't think anything else, and I was like, should this cross the membrane through diffusion? You should say, no, it shouldn't cross the membrane through diffusion because it's polar, right? But obviously living things need a ton of water. And so our cells have dedicated protein channels called aquaporins that are used just to move water, right? So it moves water in, it moves water out. All the cells have them. They have a lot of these. So water will always move according to the concentration gradient. Right? So water will always move through diffusion's principles into or out of a cell. So when we're talking about osmosis, usually we're actually talking about the concentration of substances other than water. Okay? And so we're looking at how the concentrations of other substances are impacting the movement of water because water can always move. Right? Whereas other substances like salt, table salt, sodium chloride, cannot because it's ionic. Right? And so if we look at my beautifully colored models here, okay, I have a few different situations set up. The blue represents water molecules and the green represents sodium chloride. Okay. The sodium chloride, again, cannot cross the membrane because it's ionic. Right? And so in my first model, I have 100% water outside the cell and 100% water inside the cell. Okay. There's going to be zero net movement of water because it's at equilibrium already. The concentration on this side is 100, the concentration on this side is 100, it's equal, right? So there's water molecules floating in both directions, but it's at a relatively equal rate, so there's no net movement. Okay. This situation where the water concentration is equal on both sides, we call isotonic. Isotonic. There's no net movement either way. Okay. Slide over a little bit. Okay. And in this model, outside of my cell, I've added some salt. Right? And so now my external environment is 75% water and 25% salt, where my internal environment is 100% water and 0% salt. Okay? So we're looking at the concentration of water, okay? And I have a high concentration here and a lower concentration here. So the water is going to move out of the cell, right? And now, if you think about this, right, concentration is not based on raw numbers, it's based on percentages. And so the concentration of water based on what I've drawn here inside the cell is always going to be 100% because there's never going to be salt moving in. Right? So no matter how many waters I take out, as long as there's water here, it's going to be 100% water. Right? So these waters are just going to keep moving out of the cell. So if you imagine that, right, the waters keep moving out of the cell. That cell is nice and full of water, right? As it's losing water, it's like a water balloon that's losing water. It's shrinking down, right? Oh, it's shriveling up, right? Not good for the cell, right? It gets all wrinkly and gross. Yeah. When we have solutions where the other stuff, in this case salt, is in a higher concentration outside of the cell, we call that hypertonic. Okay, hyper I think of it as being a lot, right? So I think of there's more stuff outside of the cell than inside of the cell, and the water will move out. Okay, it shrinks up our cell. Right. And then we have the opposite situation. I have salt inside my cell now, and I've left there to be no salt outside of my cell. Okay, so I have 100% outside water concentration, 70% inside water concentration. Opposite story is going to be true. The water is going to move from out to in, and it's just going to keep coming in, right? And I got my water balloon again, and as I add more water, it gets nice and big and full. Eventually, if I add too much water, that cell is going to lyse, right? And that can happen. So like if you drink too much water, you can actually kill yourself with it. You can lyse all your cells, you can make them blow up, and then you die, which is not good. No, it's not good. Right? You don't want to die from drinking too much water. Yeah? So that's osmosis. Right? There's the story of like water potential and doing maths for how to calculate how much the water is going to move. I'll do a different video on the maths for that because I don't want to fit it in here. Okay. And then finally, the last mechanism I need to talk about here before I'm done talking is endo and exocytosis. Right. And so endo and exocytosis are what cells use to move really big molecules or large groups of small molecules. And so instead of trying to cross the membrane, through the membrane or through a protein channel, the cell will actually extend its membrane out around the substance, surround it in its membrane, and then suck it in if it's endocytosis, right? So endocytosis wraps a outside substance in a membrane and then brings it in. Okay? So when you wrap up that substance in that membrane, you're creating a vesicle. Right? And so endocytosis will create these vesicles inside of the cell full of whatever they sucked in. Right? And so if it's phagocytosis, you're bringing in some kind of large solid particle. You can think of this as a cell eating, right? It's eating these things. If it's penocytosis, you're bringing in some liquid, so it's like the cell's drinking some stuff. Right? And then you get really cute little receptor-mediated endocytosis. So you get these little cell receptors here, and they can grab a bunch of the molecules, and once they're full, the vesicle will be formed and suck it in. So this is how the cell can collect a lot of one thing. Right? And those are all endocytosis, bringing the substance in to encapsulating it in a membrane. Okay? Exocytosis, similar idea, but go in the opposite direction. Okay? This is how you get rid of either a lot of stuff or big stuff. The cell wraps the stuff up in a membrane at the Golgi apparatus. That membrane will become fused with the cell's membrane itself, right? So the ball comes up, clicks, and then it kind of opens like this outwardsly, and all that material gets released out into the extracellular area. Right? Both of these mechanisms require a fair amount of energy because moving the membrane the way that it needs to be moved does require energy. So these are technically types of active transport. Cool. That's it. That's definitely it. Bye.